Hello, everybody. My name is Corbin Bauer, and today I'm going to be talking to you about our anti-cancer agent, cyclophosphamide. Cyclophosphamide is one of those alkylating nitrogen mustard agents that we commonly use in the treatment of a various variety of cancers, and today I'm going to talk to you a little bit about it. Cyclophosphamide has a couple different trade names, but is oftenly referred to as simply cyclophosphamide, which is how I'll be referring to it throughout this entire presentation. You may have heard Professor Whalen talk to it, refer to it as a cyclophosphamide, but most commonly in the clinical setting, we do just call it cyclophosphamide. There's a couple different trade names that are used for it, however, some of them being Cytoxan, Endoxina, Neosar, Procytox, and Revimune. Cytoxan, however, did lose its FDA approval and is no longer really used. The IUPAC name for cyclophosphamide is an NN-bis-2-chloroethyl 2-oxo-132-lambda oxazophosphanan 2-amine. It's a very complicated name, but once we get to the structure in later slides, I will explain how the name does correlate with the structure itself. Cyclophosphamide was first discovered in 1958 by the Osta Work Company in Germany, which is now called the Baxter Oncology Company. It's one of the largest researching companies in the oncological field and is one of the leaders of the world in terms of that research. It was discovered by a man named Norbert Brock as part of the Osta Group, and this was part of a 1,000 prodrug preclinical trial that was testing for phosphoramide mustard prodrugs. Phosphoramide mustards were first discovered and synthesized in 1954 by Friedman and Seligman. They had discovered this phosphoramide mustard and its ability to work as an anti-cancer agent, but unfortunately, due to its toxicity, it was not able to be easily ingested. And so this prodrug trial began, and Osta Work Company tested over 1,000 drugs and determined that cyclophosphamide had the largest therapeutic effect, and thus they went with it. Now, it was only a short year later in 1959 that cyclophosphamide did get its FDA approval, and it was approved both for oral ingestion and intravenous injections. The oral ingestion was in the form of pills and not any sort of liquid or other form. Cyclophosphamide, as I stated earlier, is a very strong anti-cancer agent that's used to treat various types of cancers. The main cancers it does treat is leukemia and lymphoma type cancers. So here's a couple different examples we have of some of the cancers that can be treated. As you can see, there's quite a few different leukemias around here. There's going to be some myelinomas and some of the lymphoblastic things. But another thing you do notice is ovarian cancer, you have breast cancer, some renoblastomas. There's quite a few different cancers that this drug treats, but it mainly does target a lot of these cancers that deal with the blood, the bone marrow, and cases like that. So here's the structure of cyclophosphamide. As I said earlier, the IUPAC name is that NN-bis-2-chloroethyl 2-oxo-132-lambda oxazophosphanan 2-amine. So if you can see here, the main structure that you notice here is the NN-bis-2-chloroethyl. This can later be broken down into a nitrogen mustard or some other component that will be useful, and we'll talk about that in probably the next couple slides about some of the different active compounds that are broken down from cyclophosphamide. So here you can see the metabolic pathway for cyclophosphamide. Some introductory statistics. About 75% of the cyclophosphamide that is either ingested or injected is going to be converted into its active and inactive products. Cyclophosphamide does undergo the cytochrome P450 enzyme reactions to add an OH onto it. If you can see here, it adds a OH in that 4' position. Some of the common cytochrome P450s that you see are CYP2A6, 2B6, which is actually the most active and highest hydroxylase activity. You also see 3A4 and 3A5, as well as 2C9, 18, and 19. Cyclophosphamide, as I stated, is then activated into 4-hydroxycyclophosphamide, which is then which is in equilibrium with this tautomer aldophosphamide right here. Aldophosphamide can then be broken down into an aldophosphamide. Sorry, not aldophosphamide. Let me get my notes. So it can go into the inactive 4-keto cyclophosphamide here, as well as the carboxyphosphamide over here, the 4-keto over here. And this carboxyphosphamide can be broken down into this phosphoramide mustard and the acreolin. And as we discussed earlier, the phosphoramide mustard is the ideal drug that we're trying to get that has these alkylating and interlinking effects. So some of the substituent results, as I was saying, you get this phosphoramide mustard, which is the active drug. You get an acrolein, which is excreted as a byproduct through the kidneys and bladder. This will have some side effects that we'll talk about later. And then you get the carboxyphosphamide, which can be broken down into a nitrogen mustard and also used as an alkylating agent. So because phosphoramide mustard is our main active metabolite, these are the statistics for the effectiveness of it. The EC50 value was 184 milligrams per kilogram, and this was studied in a mouse model through IPN injections. IPN is just intraperitoneal, which means just in that pocket of space right near the lower abdomen near the legs. So some of the binding activity that we see from the phosphoramide mustard is you see a guanine cross-linkage. It acts as a methylating agent and an alkylating agent. So with the guanine cross-linkage, much like you guys would see in the thymine dimers, but it's a little different. Thymine dimers cause the two thymines of one strand to bind together so they cannot be translated. Whereas this guanine cross-linking, you have two different guanines and opposing strands that are cross-linked together through this drug so that when the DNA goes to unzip to attempt to be replicated or translated, it cannot fully unzip due to this cross-linking. Then you also have the addition of the methyl groups, which can inhibit the correct utilization of base pairing, which causes mismatching in the DNA so it can hopefully get noticed and broken down. As for alkylating agents, there's a couple different things that these alkylating agents do. The primary function is to add alkyl groups to the DNA bases, so it can trigger these repair enzymes to replace these alkylated bases by fragmenting the DNA itself. This is a hope to fragment and destroy this DNA so it can no longer be replicated or translated. As I already stated, you have DNA damage via the form of crosslinks, which can be these thymine dimer crosslinks or these guanine crosslinks across strands. And then as well, you also have the introduction of mispairing of nucleotides, which is forcing mutations onto the DNA in the hopes that these repair enzymes will see it and degrade the DNA. Here's some of the side effects that we see of cyclophosphamide. Some of the notable ones are the nausea, the vomiting, the hair loss, the loss of appetite, the ulcers in the mouth and the tongue. As I talked about earlier, the acrolein, it can actually, it's excreted through the kidneys and the bladder. And so you'll see this buildup of acrolein, and it's an inflammatory agent that can cause irritation and inflaming of the bladder. So as a result, you can see some blood in the urine. There's going to be some stomach discomfort and some pain while urinating. And then some of the more major side effects and severe side effects is you do have an increased risk of developing a new cancer as well as a slow healing of wounds. Specifically in the pediatric hematology and oncology clinic that I work with, we see a lot of our patients presenting with the nausea and the vomiting and the hair loss, as well as some blood in the urine occasionally. But we do often prescribe medications that can go along with this to reduce these symptoms. So cyclophosphamide stands out from the rest of its class in three main categories: its versatility, its potency, and its permeability. Cyclophosphamide is a much more versatile drug than the rest of it in this class because it can be further metabolized from just a regular active product. As I showed earlier in the metabolic pathway, you can go into a regular nitrogen mustard, or you can go down into this phosphoramide mustard. Either way, there's two different agents that can be used to act on cancer, which is a lot more than you'll see with most drugs that just break down into one agent. As for potency, it is one of the most potent anti-cancer drugs, and unfortunately that does include immunosuppressing. It's a double-sided sword. You do have an increased rate of destruction of cancer cells, but you do also have an increase in the immunosuppressing capabilities. And then lastly, the permeability. As I stated earlier, most of the cancers that this targets is blood-related cancers, bone-related cancers, and small soft tissue tumor-forming cancers that can present in various parts of the body. And so cyclophosphamide needs to break down into this phosphoramide mustard that can very easily make its way through the bloodstream to different parts of the body so that it can target these cancers that may present in different parts of the body. Not every bone will be affected the same. Not every tumor is going to be in the same spot. So cyclophosphamide breaking down into this phosphoramide mustard is necessary so that it can reach all those aspects of the body that these different cancers affect. There is not much known about the pathways of resistance for cyclophosphamide. The few developing theories that we have is that the alkylation effects of causing mismatches in the DNA base pairing and the guanine cross-linking and all of that are most likely the pathway resistance is these DNA methyltransferases, specifically the O6-methylguanine and non-AGT repair mechanisms. They're going to start to recognize some of these mismatches that cyclophosphamide is directly trying to create and actually fixing them so the cancer cell can continue to replicate and translate more RNA and DNA. So, the O6-methylguanine can break apart those guanine cross-linkings, and the non-AGD DNA repair can fix some of those mismatches that we're purposely trying to create in the attempt to destroy the DNA. And what it does is it just simply fixes it so the DNA can continue to have those replication cycles, which is ultimately what we're trying to avoid. That'll be the end of my presentation. Here's some of the sources I have. I have citations for all the sources except the PDF on the bottom. Thank you all for listening, and we'll see you next time.